Okay, let's have a class today, and we will talk about the Endospore stain. And the fact sheet right here is encoding the Endospore stain. And also on Thursday, next week, Tuesday, we will do mixed culture and streak plating. Okay? So let's talk about the Endospore stain, what it looks like. Well, that's lab 4 Endospore stain. Okay. First of all, you want to mention, we will talk about in the lecture already, not all the bacteria has endospore. Basically, it's bacillus and clostridium. Those two major genera of the bacteria has endospore. The bacillus today we were using is Bacillus cereus. And another one is Bacillus subtilis. Okay, Clostridium, there are multiple species names, including Clostridium tetani, Clostridium difficile, Clostridium botulinum, and the Clostridium perfringens. And we will mention this in the lecture. Clostridium we cannot do in the lab because this is biosafety level 3, and the bacillus coming from soil, biosafety level 1. So today we will pick bacillus, we will focus on Bacillus cereus. So that's something we want to say. Second, endospore usually it's forming in a stress environment, which means you have like a acid, cold, UV light explosion, poor nutrition, all kinds of the stress environment, the bacteria will have forming endospore. It's a protection structure. It's not have genetic transfer function. So we also call it a no productive function. What that means, you have a mother cell has endospore, does not mean the daughter cell will automatically generate endospore. And the daughter cells has also to be in a harsh and stress environment. Okay, now what's the endospore looks like? The structure looks like a little complicated, but we could explain to you a little easy. So there's a multi-layer, okay? We're going to draw lots of layers. It could be more complicated, it could be more easy. We're going to draw multi-layer. Maybe just draw like that. So outside, this is called the exporium. Then you have, this is called coat. Then you have cortex. Okay, then you have, this is, we call it cell wall. Okay, then you have, this is inner cell membrane system. But more important is this core area. Why bacteria, the endospore, can survive in a stress environment? Because in the core area, there are two major chemicals. Number one, calcium DPA. Calcium dipolyltic acids. The second one we call small acid soluble DNA binding protein. Diproconic acids, okay, not lic, conic acids. Calcium diproconic acids. Let me double check the spelling. I want to make sure that spelling is correct. Calcium diproconic acids, right in the middle. Okay, conic acids. Okay, so that's some structure. Now we also want to say, we mentioned in the lecture already, why we want to do the Endospore stain. We want to know, first of all, presence, absence of Endospore, which means, presence, which means, is there Endospore and where is the Endospore? Second one is the location. Because the location is genetic dependent. And we talk about in the lecture already, could be right in the center. It could be on the edge, which is terminal, like you go to the airport, and it also could be between center and terminal. We call it a subterminal. So what you see today, Bacillus could be subterminal and the Clostridium could be terminal. Some of the species could be in the center. Okay, so that's the location. Now, number 3, we also want to say something very, so that's 3, that's 4. Number 4, very important, the staining will be a little bit different. Because in those small standard structure, we call it very rigid. Okay, very tough. Therefore, you cannot use just a basic dye like gram stain to do the staining. So you need some special dye. The special dye is on your bench. So Rion can prepare for us is a very green one. The green one we call it a malachite green. That's a very special dye. Another thing we also mentioned in the lecture, because the basic dye cannot go inside. So what we have to do, we need to use a very special treatment we call heat steaming. Now heat steaming, there is 2 methods to do. One of the methods will be using water bath. So what happens is if you have a water bath right here, like at the back, the water bath we already set about at least 70 degrees Celsius. And then you have the heat gonna come in, So the heat steaming is from the back, okay? Another fun stuff what we could do is some of you gonna work with me. We will put a glass slide on the rack. Then we put a Bunsen burner on the top. So this is Bunsen burner. And we put a Bunsen burner back and forth to do the heat steam. The heat steaming is about 5 minutes. Now, one more thing I forgot to mention. The malachite green, using the water bath, you only need to add right in the middle. Okay, the malachite green, you right at the middle is enough. But if you're using the methods, what I'm doing for the Bunsen burner, you have to flood it, which means the dye has to flood the whole surface. So we call it a flooding. That's not just a couple of drops. The dye has to be for cover the whole glass slides. Otherwise, the heat steaming, the heat will not be averagely covered the surface. Okay, so this is some of the introduction what I have. one more thing, because endospore is a protection structure in the stress environment, that is also takes time to grow. So endospore stem, we have to use old culture. How old it is? Basically more than 72 hours, generally speaking. But the species what we are doing today is Bacillus subtilis. This guy had a very good endospore forming capability. We only need to grow 24 for 48 hours. It will forming a very good endospore. But basically, generally speaking, different from gram stem using a very young culture, the endospore stem, you have to use a very old culture, usually 472 to even 96 hours. So it took about 3, 4, 5 days to form in the endospore. Okay? Okay, next we're going to do a procedure, also a lab procedure. So how are we going to do that? Number one is smear preparation. So we will be doing only once. So you guys draw a big drop, add the bacillus there, BS, Bacillus subtilis. Okay, now we're going to do the same thing like before. That's called smear preparation. So the smear preparation, exactly the same like we did before. We label, flip it over. Then we add water, a drop of the water, and we run the slants. We're going to pick the bacteria, go in there, and we make a big smear. Okay? And of course, you're going to have to do air dry, 3 to 5 minutes, followed by heat fixing. These are the same thing. Now remember, this is heat fixing. Is for inactivate enzyme for the bacteria and sticky onto the surface, and this is heat steaming. That is different. But we still have to do the heat fixing to prepare the smear. Okay. Now number 3, you're gonna add a dye. So today's group will have 2. If you are working with real, the malachite green is right on the middle. You're gonna add right in the middle, okay? Couple drops, cover the circle. 2 groups work with me. The malachite green has to be covered the whole surface. That need a lot of malachite green. It's flooding. Okay? Then we were using heat steaming 5 minutes. One group will do water bath, and the other 2 group will do is Bunsen burner. Now, once you do this, be very careful. The glass slides will be very, very hot. So you're going to be cooled down a little bit. This cooling down is important. And then rinse. And we do counter stain with saffroning. So what you're going to find today is still using 10x, find the specimens, and with 100x and oil immersion. You should see, based on our previous experiences, is UCC background is relatively very red. Okay, that's saffron in color. So you should see a bacteria, a lots of the malachite green like that. And what you should see use a big bubble circle right here. This is an Endospore. We will see what it looks like. This is what, before we find it's a big bubble there. Looks like one of the slides in our textbook. Not like really the one you have a green color. It looks like it's a big air bubble. So what we're going to do is that, since you already know the smear preparation, everybody get one glass slides to prepare the smear until heat fixing. Then we will have two groups. One group is going to working with me. We're going to do the flooding and heat steaming for 5 minutes. Another group work with Rio. We'll just do the water gas. Okay, so that's will be. We'll do, these two groups work with me, we'll do the steaming, and the other two groups will work with Rio, we'll do the water bath. Okay?
Make sure you guys write your bench